This is a presentation on Bolitoglossa Striatula, also known as the kukra climbing salamander, a salamander found in parts of Costa Rica, Nicaragua, and Honduras. So, morphologically, the characteristics that were first observed by G.K. Noble upon identifying the holotype of this species was brown dorsal and lateral stripes, lateral on both sides, and webbed feet, more webbed than other salamanders of the genus. There's also a pale tan dorsum on its back and a large amount of fine brown streaks on the head. In addition, Noble also observed a elongate body with, as you can see here, a cylindrical tail. In later analyses across a larger sample size, Saunier observed a sexual dimorphism of the snout ventral length, 51 millimeters to 58 millimeters when you compare males to females of this species. Morphologically, we see that this Bolitoglossa salamander has the smallest adult skull out of all the other salamanders found in the genus. It comes with no fusion in some of the bones. You can see up in the center here, I'll have the cursor, actually yes, the center here, that this bone, the frontal bone, hasn't fully fused, and there's only a single point of contact. And there's also a smaller, relatively small amount of ventral teeth compared to other salamanders of the genus. And here's some measurements that were observed in the holotype. This species is located from northeastern Honduras to southeastern Costa Rica, and there's a large variation in elevation from sea level to 1,380 meters is the highest it's been observed. This salamander goes through direct development, and it emerges from the egg as a fully developed lungless adult salamander. It remains in arboreal and terrestrial habitat, as observed originally near a plantation in the original paper that it was described in. It prefers moist forest and rainforests that are close to fresh water, and it's often found camouflaging against yellow leaves, tall grass, and reeds, owing to a tan dorsum. Currently, it's classified as least concern, but there's decreasing population, and its habitat is currently facing issues from agriculture and logging, as well as urbanization. These animals are sister to Nanotriton and Myomandra, as derived from mitochondrial DNA samples in museum specimens, and it's very similar morphologically and molecularly to the Mexican climbing salamander.